Chemotherapy. When the cells divide, they go into the very well-controlled process so-called cell cycle. First of all, the cell needs to accumulate enough materials to make its copy. So this step is what we call G1. Here G is a gap. So first the gap is the G1. When they accumulate enough material, then they start to synthesize the DNA. So this phase is called as a S. It's a synthesis of DNA. When the DNA is duplicated, the synthesis is completed. Then it goes into another gap step to check whether everything is okay to divide. So this step is called as a G2, gap 2. And then finally, the mitosis process starts. And mitosis, first of all, the DNA should be aggregated to form chromosome. This is a prophase. And then those two sets of chromosome is aligned in the middle of the cell. So this is a metaphase. And then each set of chromosome is moving to each end of the cell. This is anaphase. And through telophase, and finally, the cell division happens. So the final step of the cell division is called as cytokinesis. Cancer cells can grow forever, so this is a replicative immortality. That's why cancer cell has an uncontrolled growth with invading other part of the tissue, so-called malignant behavior they have. That's why most of the chemotherapy drugs work by attacking the cell division, so effectively targeting the fast-dividing cells. Fast-growing cells, they should go into the cell division cycle, and this cell cycle has several checkpoints. First checkpoint is before DNA synthesis. So this checkpoint is about whether the cells collected enough material to make a new set of DNA and also to make a new body of the cells. Second checkpoint is the G2 checkpoint. This is after DNA synthesis. So this is to confirm the DNA synthesis is properly completed or not. And another checkpoint is M phase, the checkpoint. This is to check whether those division of two sets of chromosome is properly completed or not. DNA alkylating agents can make a covalent bond to DNA structure. So DNA alkylating agents such as mustard reagent, this compound has a good electrophilic point, and this can be attacked by nucleophile of a DNA base. And then multiple reaction here can make cross-linking to different DNA strand. The cross-linking can be interstrand or intrastrand. Interstrand means this is between two different strands. Intrastrand is from same strand. And typical reagents for this is nitrogen mustard and cisplatin. Second class of chemotherapy is anti-metabolites. To synthesize a new strand of DNA, the cells should have enough the purine building blocks and pyrimidine building blocks. So the purine analog 6-MP can block the synthesis of purines, and pyrimidine analog 5-FU can block the synthesis of new pyrimidines. For the pyrimidine, folate is also important, the metabolite, and if anti-metabolite is added, then pyrimidine synthesis can be blocked. So this kind of purine analog or pyrimidine analog or anti-folate, they can interfere with S phase completion. So these reagents interrupt the S phase. For DNA replication, the two strands of DNA should be opened, and helicase is taking that part of catalytic activity. Another enzyme, topoisomerase, is also important to release the strain from the supercoiling. There are two kinds of topoisomerase, type 1 and type 2. Type 1 cleave one strand of DNA, and after releasing the strain, it reconnects. Topoisomerase 2 cleave both strands, and then later they reconnect both of them. Topoisomerase 1 inhibitor, such as topotecan, interrupts S phase because that interferes with DNA synthesis. Topoisomerase 2 inhibitor, such as doxorubicin, that acts at the end of DNA synthesis. That's why this interrupts S or G2 phase. The last kind of chemotherapy is anti-microtubule agents. When the cells enter M phase, now two sets of chromosomes is aligned, and then microtubule is attached to the chromosome and then pull over to each end of the cells. So here the microtubule is an important machinery for cell division. Microtubule destabilizer, such as vinca alkaloid, it can prevent the metaphase formation. To make the metaphase, chromosome should be aligned in the middle, and the microtubule should attach to the chromosome. Now, this destabilizing effect can prevent the proper formation of a microtubule. Interestingly, microtubule stabilizer, such as taxane, this is also harmful to fast-dividing cells. After metaphase is formed, now the two sets of a chromosome should be segregated. Now, overstabilizing of microtubule can prevent this step. That's why taxane, such as paclitaxel, can be also important anti-cancer drugs. So, for the microtubule, destabilizer or overstabilizer, both of them are important chemotherapy reagent. So let's just summarize those chemotherapy agents. First set was a DNA alkylating agent, which can make DNA structure unstable. And anti-metabolites, it blocks the synthesis of purine and pyrimidine and interfere with the DNA synthesis. Topoisomerase inhibitors also interfere with the DNA synthesis. Type 1 is in the middle of DNA synthesis, type 2 at the end of DNA synthesis. Vinca alkaloid, the microtubule destabilizer, is blocking the entry into metaphase. And taxanes, microtubule overstabilizer, they can block the progress from metaphase into anaphase. So these are the representative classes of chemotherapy agent.